Alright, Dr. McNeil, Alex Shaw behind the camera, fixing the ends of linear chromosomes. Okay, why does this matter? Well, all important organisms that you or I care about, like people, or ponies, dolphins, dogs, cats, marmots, platypus, any of those cool animals, any eukaryotes, have linear chromosomes. Okay? So, this is not bacteria. This is only eukaryotes. And bacteria aren't going to have this problem because they don't have a linear chromosome. So I'm going to explain to you the solution that all higher animals use to fix the problem of having linear chromosomes. Okay? So what are we going to use to do it? We're going to use an enzyme called telomerase. What we're going to find out, what I already know, but what you're going to find out is that telomerase is a protein RNA complex, right? And it has one super important activity, and that is it is a reverse transcriptase, right? ACE enzyme. It's an enzyme that does reverse transcription, okay? In other words, its main enzymatic activity is RNA dependent DNA polymerase activity. And telomerase is required for life in all higher animals. Okay? So, what's the problem here? Well, here we've replicated nearly an entire chromosome. We're on our last replication bubble. Okay, so we have replication 4 moving this way. Leading strand of this is going to the left up here, but ultimately by the time we get to the end of our 4, the lagging strand is up here because it's being made from right to left, despite the fact that our fork is moving to the right. We've just reached into the chromosome, so I haven't redrawn the end of the replication fork, because that doesn't exist, because we're at the end of the chromosome, the very end, okay? So what's going on? Well, as usual, we have DNA polymerase 3. Remember that DNA polymerase 3 is a homodimer, right? It's got one DNA polymerase moving to the left on the lagging strand, DNA polymerase moving to the right, doing synthesis of the leading strand. Okay. Now hopefully you can see right here that on the bottom, the leading strand, there's going to be no problem. So as our fork moves from left to right, DNA polymerase 3 is going to chug all the way down. I'm not going to move the DNA polymerase, but it's going to chug all the way down and keep going until it reaches the very end of the chromosome. And then it's going to stop. So we now have a new 3' end of this chromosome. So with the leading strand, there's absolutely no problem. No problem with the leading strand. What is there a problem with? Well, there's a problem with the lagging strand. And why does this problem come about? It comes about because of exactly what DNA polymerase can and cannot do. DNA polymerase, you should already know, cannot start new strands. It can't start new chains of DNA. It never could, never can, and it never will. Right? You know that from lab, from doing PCRs, you need primers. You know it from doing regular DNA synthesis inside of a replication fork, like we went over in the last video. Right? You also know that DNA polymerase must have a 3' to 5' template. It doesn't randomly write nucleotides, it only writes nucleotides using its template. So these are our two main limitations of DNA polymerase that we care about. Okay? So, why does this matter? Well, it matters because DNA polymerase 3 here can only extend a chain. It cannot start new strands of DNA. So we have primase lay down an RNA primer in a 5' to 3' direction, and the last primer laid down here is shorter than the chromosome by, in this example, 12 bases, but it could be a lot more than that. So primase has laid down its very last primer. DNA polymerase 3 is extending that primer from the left, from 5' to the 3' direction. Okay, that's awesome. But what happens? Well, right now, we're stuck with single-stranded DNA. You know that our genome isn't made up of single-stranded DNA, right? Single-stranded DNA looks a lot like a viral genome, and that's eligible to be broken up by your native enzymes, by exonucleases, okay? So we need a mechanism that overcomes this single-stranded DNA, and the other thing we need to do is when this bottom strand, this new strand of DNA gets copied, it would all of a sudden be shorter than the mother strand. That doesn't sound like a big deal. Yeah, here we lost 12 bases. Imagine you're a stem cell in the gut, and your job is to divide once every 24 hours for 75 years. Right? If you do that and you lose 12 bases every time, how many days are in a year? 365 times 12, you're losing 3,000 bases a year in that division, essentially. And over 10 years, you've lost 300,000 bases, and very quickly you begin to delete genes on this chromosome. Okay? So it's not acceptable to lose any link on the end of a linear chromosome. You just can't do it, can't afford to. So what happens? Well, this is where telomerase comes in. Remember, telomerase is an RNA-dependent DNA polymerase, and it's a protein and RNA complex. So I'm going to erase a couple of these labels here. The telomerase complex is freaking huge. It's many, many proteins, many more than we're ever going to talk about. I'm going to erase your carriers there. And the telomerase complex, made up of more than a dozen proteins, it comes in, and the first thing it does is binds to the lagging strand during DNA synthesis. So that's a telomerase here. Okay? Second, you might wonder how it binds to the lagging strand, how it knows where to go. It uses the telomerase RNA to localize itself. What does that mean? Well, that means that in telomerase, there is a 3' to 5' telomerase RNA. And telomerase RNA is about 500 base pairs long. Different sizes in different eukaryotes. What's important here? The telomerase RNA is complementary to the repeat units at the end of linear chromosome. So these are telomerase repeats. And they didn't get here by accident. They got here through complementary base pairing. C, 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 A. U, C, 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 A, A. U, C, 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 A, A. U, C, 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 A, A. It got here through complementary base pairing with the linear end of the chromosome. Okay. So, what else is telomerase? Well, telomerase is RNA. It's also proteins. It's a reverse transcriptase. Okay? So what happens? Telomerase is here. This whole complex is telomerase. Okay? And telomerase uses its reverse transcriptase activity to extend this 3' OH group off the end of this linear chromosome, because it's a DNA polymerase, it extends this thing. It writes DNA, A, G, 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 T, T, A, G, 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 T, T, A, G, 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 T, T. And it does this over and over and over again, times about 50 times, right? And it's behaving itself like a DNA polymerase is supposed to do, because it's not starting a new chain, it's extending into the linear chromosome, and it's using a 3' to 5' template. It's using the telomerase RNA as a template, okay? Telomerase RNA is our template. So what's it doing? Well, it's extending the length of our chromosome. And so as telomerase writes more and more and more and more and more DNA, what do we end up with? I'm getting rid of the telomerase RNA now. And we end up with a much longer chromosome than we started with. So our previous chromosome only went to right here. Our new chromosome now ends several hundred base pairs away, okay? What this room does is allows for primase isn't orange, primase to come back and write a new 5' to 3' RNA primer, which still technically isn't at the end of this chromosome. There's still a little bit of single-stranded overhang. But it allows primase to come in, lay down a 5' to 3' primer. As this fork, the DNA replication fork, continues to move to the right, this DNA polymerase 3 molecule eventually is over here on the 3' end. So this whole thing, all this DNA polymerase has translocated to the right as it moves down the linear chromosome in the direction of the fork. So now this DNA polymerase 3 can rewrite new DNA in a 5' to 3' direction all the way back to the previous primer. And at that point, this looks just like a regular replication fork. There's nothing special about it now. Okay? So, 3. Telomerase 5' to 3' DNA pole activity. Writes telomeric repeats, extending the 3' end of a linear chromosome. Okay? All this extra DNA simply allows for a new priming event outside of the old chromosome boundary. Okay? So, what are we left with? We're left with a space for DNA polymerase 1 to come back, bind to our RNA-DNA hybrid, our Okazaki fragment, to use its enzymatic activity, its 5' to 3' exonuclease activity, to exclude the RNA primer and to fill in DNA in the 5' to 3' direction. Then we can have DNA ligase come in and ligate the connection between this new DNA and the DNA we'd already laid down. Same thing happens here. New DNA comes down, DNA polymerase 1 writes to there, filled in by DNA ligase, everything's happy. What's still our problem though, is we have a slight bit of single-stranded DNA at the end of our linear chromosome. Okay? So the last thing telomerase does is probably not really remarkable. Telomerase remains bound to the end of the chromosome. So telomerase stays right here at the end, guarding all of this single-stranded DNA and maintaining the integrity of this chromosome from being destroyed by enzymes. So, what do we learn here? We learn it's almost exactly like a regular replication fork, except that we extend the 3' end of the lagging strand only, extend the 3' end of the lagging strand using telomerase's RNA-dependent DNA polymerase activity so that we could reprime and lay down new DNA coming back. Simple as that. Why is that important? Why is it important? It's important because if we don't do that, your chromosomes get shorter, and some gene over here that's required for, say, heart function gets deleted if we don't.